Hello everyone, you're welcome back to my YouTube channel. My name is Mrs. Sandra. Today we have an interesting topic in medical microbiology, bacteriophages. Did you know that just like every human being, bacteria also has friend enemies? Who doesn't have one? We all do. Interestingly, these friend enemies called bacteriophages are viruses just like the HIV virus, which are obligate intracellular organisms. When I mean obligate parasites, of course, they need a living host to survive. And interestingly, this group of viruses have chosen bacteria to be that host. This actually works for us as microbiologists. And today we'll be talking about how to isolate them in the lab, how to cultivate them, how to pick them up from the environment, how to manipulate them to our own use. It's going to be an interesting class, so stick around and join me, okay? It's important to note that bacteriophages are specific viruses. That means they need a specific receptor on the site of bacteria to attach. They don't just randomly attach to any bacteria. Bacteriophage requires a specific receptor on the site of a bacteria, like lipopolysaccharides on a gram-negative organism, to attach. So they are actually specific in action. That's what I'm trying to say. They have specific likes, like just like when you like someone, you get attracted to the person. That's how bacteriophages work. And just like every other virus, I also wanted to mention that they have two stages. They have the lysogenic phase and the lytic phase. We know that the lysogenic phase, in this relationship with a host, the bacteriophage gets into the cell, attaches, enters the cell, begins to secrete its DNA, but do not kill the cell, do not kill the host. It creates a mutual relationship with the host, and at the end of the lysogenic phase, which you've already been taught, at the end of this lysogenic phase, the bacteriophage attaches its chromosome to the DNA or RNA of the bacteria in question. But in lytic phase, the bacteriophage, what's the right word for this? The bacteriophage storms into the host organism. It hijacks the host system and destroys it. It inserts its DNA or RNA, begins to multiply, and eventually bursts the cell, releasing its offsprings, if you'd use that word. So that's basically what happens in this too. It works just like every other virus, how it invades a host system. Then interestingly, let us go straight to what we'll be talking about today. Where can we find bacteriophages? Okay. It's important to note that the rule of thumb for finding a bacteriophage is to look for where its host exists. Remember, these are viruses. They cannot just live on their own. It's just like when you want to find an ant, or these ants that eat sugar, you just go straight up to the sugar. That's what works with bacteriophages. The rule of thumb is to look for them where their host exists. That's where you find them. And most times, water is a very popular sample collector for bacteriophages. The bacteria can be found in water. Water is usually a popular sample that is used when trying to isolate bacteriophages. Now, the water sample is being collected. Then the bacterial choice, since you know that the bacteriophage is specific in action, is mixed with this water sample and left for a few moments. I'm just giving a general overview. Of course, I'm going to give a detailed overview on our slide. They are now mixed together, and after a while, they are being centrifuged, and centrifugation is being carried out to bring them down the important sample that we want. Then we now filtrate them. We know that viruses are very small microliter, so we set the filter to the microliter we want, which only the viruses can pass. Then we filter. Then after that, we prepare a soft agar gel which contains the microorganism, that's the bacteria and the virus. Then before that we've already prepared TSA agar. Then we now pour this sample containing the virus and the bacteria as a soft gel on top of the already prepared TSA agar. Then we allow it to grow. After, you set it to grow after 24 hours, when we come back, we'll find out that there are circles which we call plaques that are shown in the plate. I forgot to mention that serial dilution is going to be carried out so that we can dilute and set and watch for exactly what we are looking for, because if you do not dilute, you're going to have a flood of plates. I know that in microbiology we need 30 to 300 CFU, but in this situation we're calling it plague forming units. So when we carry out serial dilution, then we pipette out our mixture, then we pour the soft agar on our TSA agar, then we allow it to grow. After the growth, we are going to notice a distinct clearing zone, which we call plaques on the plate, and this is how we identify. This is what we identify as presence of the viruses, and we call it plague forming units. And just like we calculate CFU, that's the same way we calculate plague forming units. Okay, let me just show you guys a pictorial view in my laptop. One moment. Okay, so this is the filtration method I was trying to talk about. And next, I think I have a picture of this, is a detailed explanation of what I talked about, which we're still going to discuss in class. So okay, this is how the plaques will come out. If you did not dilute, you're going to have a large growth here, but when you dilute, you can see plaques showing up in the plate. Okay, so the next thing we're going to be talking about is the purification of these organisms. I also forgot to mention that we use chloroform while still trying to isolate them to kill off any, we have filamentous phages, so we use chloroform to kill off those phages that remain intracellular. We do not actually like lysogenic phases that do not move to lytic phase. So to eliminate any phage that is still in lysogenic phase, we use chloroform to kill them off, mostly filamentous capsids. That's what I'm just trying to say. Okay, so let me just run down. I'll still see you guys in class though. Let me just run down. Okay, I just gave you guys a general overview. And purification of these phages, they are purified by, we actually pick them up from their plates. That when you've already created the plague, when the plague has already been set on the soft agar, we can use what do we call it? We can actually use a loop. We can use a loop to dig out from that plague a soft agar from the growth you already seen, that area where you already seen the plague. You pick it out and you put it in a paper. Oh god, I wish I could show you guys. Okay, one moment. Okay, this is what I was trying to say. You use a loop, inoculating loop, to dig out the particular area on your plate that you're trying to get your virus from. Then you put it into a 25% Ringer solution. That's what they actually use. Then you mix it, and then after that you can now keep it kept in an ambient temperature. Okay, the purification is going to take a long process. I'll be teaching you guys that one in class, but I hope you guys understood what I talked about, phages, and where we can get them from the environment. And I forgot to also mention that it's important to maintain aseptic conditions when you're carrying out this process in the lab. Okay, and I also forgot to mention that there is also something called a spot test. Because this one I explained is the plague assay. Instead of carrying out doing a soft gel and preparing an already prepared plate, you can just prepare a plate, grow your microorganism on it, then using your already centrifuged and filtered sample containing the virus, you can just drop them on your plate, and of course you still see plagues. Just that one is used to find out the presence of these viruses. It's not actually used to count them. The plague assay is actually used to count them. Of course, thank you guys very much. We're discussing this and everything else in class. You can drop your questions for me. I'll see you guys in our next class. Thank you and bye.